Hello, and welcome to today's webinar, Reducing Breast Cancer Health Inequities Across the Cancer Control Continuum. I am Dr. Barbara Malkus, Board President for the Massachusetts Breast Cancer Coalition, and I'd like to thank you all for joining us today. Massachusetts Breast Cancer Coalition is dedicated to preventing environmental causes of breast cancer through community education, research advocacy, and changes to public policy. I am pleased to welcome today's speaker, Dr. Mary Beth Terry. Mary Beth Terry, PhD, is a distinguished cancer epidemiologist with over 25 years of experience in breast cancer research, focusing on genetics, epigenetics, and biomarkers of environmental exposures. Since July 2024, she has served as Executive Director of Silent Spring Institute, leading studies on environmental risks during key developmental periods, especially in high-risk communities. Dr. Terry has contributed significant insights into early life exposures and breast development through her role as co-developer of the Legacy Girls study and over 450 scientific publications. In her previous role as Director of Community Outreach at Columbia University's Cancer Center, she built networks to address health disparities and support policy change. She holds a PhD in epidemiology from Columbia University and an MA in economics from the University of Washington. Dr. Terry, such a pleasure to welcome you to today's webinar.
Well, thank you so much, Dr. Malkus, and thank you so much to the MBCC, who's really been leading the way in making sure that the community knows about environmental causes of cancer, as well as helping resource and start these critical initiatives. So thank you all for joining today. So I'll be addressing breast cancer health inequities. And so in order to do that, I think in particular, it's really important these days to really start off with what are the terms. And I know a lot of terms are being conflated these days. DEI is being conflated with health inequities. And I really want us to start on the same page, which is at the grossest level of health inequities is something that Dr. Martin Luther King informed us of over a half century ago, which is of all the forms of inequality, injustice in health is the most shocking and inhumane. And the WHO has many resources on health inequities that exist around the globe. Of course, life expectancy is really the grossest one in terms of the biggest inequity. And that's just based purely on where a baby is born. There's as much as 34 years of difference in terms of life expectancy. And I'm sure everybody on the call today will know that that is really what we're talking about when we're talking about inequities. So the National Cancer Institute has defined the difference between health disparities and health inequities. And so disparities broadly are differences that you see in incidence, prevalence, and mortality in the burden of cancer. And health inequities are disparities, but specifically they're disparities that exist because these health outcomes are unfair, unjust, and avoidable. And those are due to systemic inequities. And we'll see by the end, they're definitely inequities that exist because of differences in environmental chemical exposures. So I'll very briefly go through breast cancer incidence trends. I know everyone on this call is very knowledgeable about them, but I think it's helpful to kind of ground us in really where we are globally. Then I'll touch upon some of the research we've been doing, and then thinking about where to go from there. So as many people know, we're in a very different place than we were as recently as 20 years ago, when very large, high-density countries like India used to have cervical cancer as the #1 female cancer. Pretty much on every continent now, we see breast cancer is the #1 female cancer. But very importantly, as the International Agency for Research on Cancer showed us, that 2/3 of all cancers that are diagnosed in individuals under 50, this is sometimes referred to as early onset cancer, are diagnosed in women. So this is one of the biggest disparities that exist in cancer. And it's one that we really need to address, because this is a pivotal time in people's lives where they're growing their careers and their families and contributing to community. So we had looked at this gender ratio recently in terms of not just what's happening now, but what's been happening for the past 20 years. And certainly what you can see for the past 20 years is this gender ratio where, again, 2/3 of all early onset cancers under 50 are really seen in women, primarily breast. On the left-hand side, this is the breast cancer trend, and thyroid cancer as number 2. And certainly there's an increase in early onset cancers. In red here, we have colorectal cancers, which are increasing both in men and women. But we can't ignore the fact that breast cancer is really the major early onset cancer still on an absolute basis. So if we look at this graph, this is global data for 185 countries, and it's looking at trends over the last 30 years. So on the colors of each dot represents a country, and the colors represent the region of the world. I'll just play that again. So what we have on the x-axis are fertility rates. What we have on the y-axis are breast cancers diagnosed under 50. And what you can see with this movement over the last 30 years is clearly a movement towards the upper left quadrant, clearly a movement to where we have lower fertility rates and higher breast cancer rates under 50. However, what I would point out is there's still substantial heterogeneity or differences across this. So if we look at, for example, countries that have similar fertility, we see as much as a 7 or 8 times difference between some countries up here who have very high rates of early onset cancer. And similarly, we can look at the rectangle down below where we see similar ranges of breast cancer incidence, but very dramatic differences in fertility rates. So again, I would point out that you have these correlation with lower fertility and higher breast cancer risk, but by no means is it the sole explanation for what's going on. And as I'll discuss in a minute, pregnancy itself is not a carcinogen, and in what we know is pregnancy and fertility rates may change the windows of susceptibility. Sorry, my computer is doing your update right now. Yes. Hang on. All right, can you see my slides now? Yes?
Yes, we can still see your slides. Thank you.
Oh, perfect. Okay. I was worried the whole computer was going to shut down. But another indicator that it's not just fertility and not just pregnancy, as many of you know, in the United States, we started registries in the early 70s. This was instituted as part of the war on cancer. And you can see now, when we look at some of our US statistics, we look at under 40 years of age as early onset, because still we do a lot of screening between 40 and 50. So we really want to make sure that this increase in early onset cancer is not driven by more breast cancer screening. By the way, mammography itself didn't really pick up until the late 90s, when you had about 80% coverage. But if we were to just look at these rates from the 70s, what you can see is a decline in fertility. These are different fertility rates, specifically live births. So this is parity going down, as we know has happened since the 1970s, and early onset cancers going up. So if you just stop there, you would conclude that maybe there is this correlation, again, between thinking about these reproductive experiences and early onset breast cancer. But if we go back to the one state in the United States that did capture registry data from the 1930s, again, showing the importance of having these cancer registries, you can see that early onset breast cancer, which is represented here by this jagged line, has been going up ever since the Connecticut Tumor Registry has been reporting early onset. and way before the baby boom, as well as after the baby boom. So again, fertility itself and parity cannot explain what's going on with these early onset breast cancer rates. So a few years ago, we published some of the registry data by molecular subtype. There's a lot of information here, so I'll just focus in on the most aggressive subtype, which is triple negative. And what you can see for each of these age groups is you can see almost a doubling of triple negative breast cancer in non-Hispanic Black women compared to non-Hispanic white women. And that's true of every age, not just early onset. And so again, many people tend to think that early onset cancers are mostly estrogen receptor negative cancers or triple negative breast cancer. But again, triple negative breast cancer, as with every other type of molecular subtype of breast cancer, occurs the most in older women. So, and luminal A, which is hormone receptor positive cancer, is actually increasing in women under 40. So these are things to be thinking about. But a major health disparity, in addition to gender being different, is this doubling of triple negative breast cancer in non-Hispanic Black women compared to non-Hispanic white women. So again, all inequities are disparities. Some disparities are also inequities, but all inequities are disparities. So what would determine the difference between whether or not something's a difference or whether or not something is driven by factors that are unjust, avoidable, and related to policy that needs to change? So here on the right-hand side, you can see mortality differences. I would point out that these mortality differences, although they're declining a little bit, are mostly stable, and they've been mostly stable for the past 20 years, and in particular in cities that have really good cancer screening data. These mortality differences exist even if mammography rates are very high. So again, screening is not really determining what are these differences in mortality, and I would argue that what still needs to be really translated is that it's across the whole continuum. So particularly with prevention and how we can think about environmental causes that may be different exposure and again, differently distributed, because as we know, environmental chemicals themselves are not uniformly distributed. And so here what I have is the cancer control continuum. So starting with primary prevention, cancer screening, we usually refer to as secondary prevention. And then once someone's diagnosed with cancer, we call that tertiary prevention. And so what I would argue is across this whole cancer continuum is that there's both a need to look within high-risk populations, and high-risk populations we'll define in a minute, but includes people that we know have very high genomic susceptibility based on cancer family history, as well as population-wide approaches. And I hope by the end of this talk, you'll be convinced that some of these studies that we do in high-risk populations are very relevant to populations that do not have high risk because of family history. And then we'll also look across the cancer control continuum for the implications on screening and treatment. So as was mentioned, I've been very interested in two major areas of susceptibility, and they kind of all come together in thinking about how the environment influences our risk of cancer generally. So a few years ago, using some of the data from the Cancer Genome Atlas and other publicly available data, researchers published this graph, which I think is very useful for all of us to think about in the prevention world, which is the red a curve here indicates the general curve for cancer incidence, which fortunately is lower earlier in life, and then it ramps up, and then it plateaus, so to speak, which most people at this point think directly mirrors this green curve, which is what we call biological aging or your biological fitness. And so this has been one of the big challenges in cancer research, because cancer occurs within us, cancer is us, and cancer, to control it, is understanding how we age, because eventually, if you live long enough, and you're lucky to live long enough, most people do have some sort of cancer based on autopsy studies. So the importance here is trying to understand when we can implement prevention initiatives. So most molecular biologists will say that cancer is 100% genetic. And what they mean by that is it either occurs because we have these germline mutations inherited from our parents, or it occurs throughout life because of what we call somatic mutations or acquired mutations. So here in brown is representing what is thought to be the acquired mutation accumulation for cancer generally. And one thing you can see is it plateaus much, much earlier than cancer plateaus, meaning that the mutation accumulation that causes cancer throughout life is more likely to occur earlier in life. And in fact, the estimates are as high as 50% of all cancer genes that are mutated through this acquired mutation, essentially how the environment gets under our skin to cause cancer, occurs before the body's fully mature, which is usually estimated to be about 25. And so that's important to know, because we also know that the breast is influenced by different periods of life when tissue itself in the breast changes in form and function. And so my colleagues at Silent Spring did this wonderful study that I told them, and here's the proof that I include in almost every talk I give, because I think it's an important slide, because this came up just the other day, and it has come up many, many times when I speak to clinicians, which is there's so many studies on the environment and breast cancer, and they're all inconsistent. So what are we supposed to say to our patients? So this was a systematic review that Dr. Brody and colleagues did, which showed, yes, there are a lot of studies. In fact, in just these 10 years alone, there was 158 studies. So we do not lack for a number of studies. But as their team pointed out, most of these studies were not specific to a window of susceptibility, meaning not specific to when the breast changes in form and function, where laboratory studies will tell us that those are the periods where the breast is most susceptible to carcinogens. And they looked across a variety of different chemical compounds, and the darker shades here indicate where most of the evidence is. So most of the evidence is in postmenopausal women during non-specific windows of susceptibility. So what I'll argue for is, again, this is important in thinking about what's driving the consistency. Because again, for thinking about, particularly in understanding environmental exposures in a non-occupational setting. So in a setting where we're all exposed every single day to hundreds of chemicals, how do we measure those, and how do we get a consistent signal when we're not looking at susceptibility? So, and as they pointed out, most of these studies have been done in animal studies, but in, the minority of these studies are done in specific windows. When we look though at the data that did exist for windows of susceptibility, we saw that the evidence was much more consistent. In fact, that also extended to organochlorine exposures, particulate matter, metals, etc. So again, when we looked at studies that did have information on when those chemicals occurred, you got a much more consistent story. So just sheer numbers of studies are not as informative as looking at the studies that actually addressed the susceptibility windows. So we were interested, and with collaborations across the country, through the Breast Cancer Research and Environment Program, which is where I had the opportunity to meet and work more with Dr. Brody. In New York, we did a study of a multi-generational study of moms and their daughters to understand how these environmental exposures were affecting breast tissue. And so the moms themselves wore these backpacks for 48 hours. We also collected blood and urine. And then approximately 15 years later, during adolescence, we measured the daughter's breast tissue and the mother's breast tissue. And for a set of polycyclic aromatic hydrocarbons that we measured, we were able to show using this device that looks a little bit like a science experiment, but we hope eventually will be approved by FDA to be used in more research studies, and particularly in young women because it doesn't involve any radiation. And what it does is it measures the chromophores in the breast tissue. And so specifically, we have these different chromophores that we were able to look at. Water, the level of water in your breast tissue maps very closely to dense breast tissue. And we do know that the amount of dense breast tissue is a predictor of breast cancer risk. We also saw in both the daughters and the mothers higher collagen levels. So again, these were exposures that were when the mother was had a susceptible window during her pregnancy, and when the daughter had a susceptible window during her intrauterine period. And then we measured breast tissue objectively. So none of this was related to recalling exposures or questionnaire data. These were all, objective measurements. And then lipid levels decrease because lipid itself, the higher those levels are, the lower your breast density. This kind of instrument too, we've used in a number of different studies now because it also measures your metabolic activity within the breast tissue, which we think is very important, particularly during the premenopausal years. So why do we want to think more about susceptibility? So again, the windows of susceptibility, we know that's when the breast is changing. But what about genomic susceptibility? And so what we know here on the right are there's many different cancer genes. And in fact, a lot of progress over the past 20 years in terms of what are cancer genes. And so here, this schematic just represents cancer genes. The size of the font indicates the prevalence. So like you can see P53 is important in most cancer organs. And then BRCA1 is much smaller here in the breast because again, it's mutated in a very small amount of people through the germline. Now it's mutated in a much, much larger level amount of people in through acquired mutations. So the same genes that are represented in the germline, which is here on the right, are the same genes that are mutated throughout life. And so in the olden days, if you will, we would do these family-based studies to find genes. And one of the studies that I've run for many years now called the Breast Cancer Family Registry, I've run with amazing colleagues around the world. We were looking for BRCA3. That's how we got funded. But what we have since used the cohort for is a number of different modifier studies to understand why some people get cancer and some people don't. Because unlike other genetic diseases, cancer, there is not a single gene for any cancer that's 100% penetrant, meaning 100% probability that you're going to get cancer. So there are some genes like BRCA1 and BRCA2 that are highly penetrant, but again, do not mean that if you have the germline mutation or alteration that you will get cancer 100%. So here's a study that I thought is really important with how we conceive of studying humans, which, it's really hard to study mice, but it's especially hard to study humans. And so what you can see here in left is these are all the genes that have been found through 20 years plus of research through all the different groups around the world. So there's over 1,200 cancer genes. And you see in pink, these are the number of genes that are mutated in the germline, so through your family genetics. And then in yellow are the genes that are mutated throughout life, so the acquired mutations, the somatic burden. And what you can see is a very large overlap between those genes that are mutated through the germline and those that are somatically mutated. There's about almost 300 more that are somatically mutated. So why is this important for how we do our designs? So here you can see this is a log plot, meaning that, again, this would be a curve, except it's presented on a line. So what you can see is what we know. And this is using the Cancer Genome Atlas data. So as you go through life, your family history in terms of your germline genetics matters less. So if your sister gets breast cancer and she's in her 70s, that matters a lot less to your susceptibility than if she were in her 30s. And so, as you can see, as we travel through life, most of us do acquire more family history because family history, again, accumulates as people age because cancer accumulates as people age. But the burden from that family history goes down. Whereas quite the opposite is true for the other types of mutations, which are much, much more prevalent. So for some cancers, we think as little as 5% are explained by these germline mutations. And so the vast majority of cancers are explained by somatic mutations. Truth be told, every cancer is an interaction between the genes and your environment. But again, these somatic mutations are much more important. And as you can see, as people age, those are the reasons that people are getting cancer, not from the germline. And so why is this important for when we're studying? And remember, I just showed you 100 studies were done on environmental chemicals and breast cancer from 2006 to 2016, because most studies are measuring women in this period of life. So most people are, when most cancer occurs, is when most people are being included in these studies. And so that has implications for these inconsistencies. So very briefly, here's how I kind of think about it. So, So the majority of non-occupational studies, and now that we have more occupations in females that we know are related to breast cancer, like active duty military women have higher early onset breast cancer than what the registry rates show for their age. Also, female firefighters have higher rates of breast cancer. Again, those occupations have been shown to have higher risk, but most studies are looking at non-occupational exposures. So the exposures we're exposed to every single day. And fortunately, again, when we're younger, we can repair some of that damage. So that's one of the reasons everybody is not all walking around with cancer, even though most of us are exposed to carcinogens every single hour, every single day. It's estimated that every 3 days somebody has a cancer cell in their body. But again, if you are able to get rid of that damage or repair that damage, that is why, fortunately, not everybody has cancer from these environmental exposures. But again, what we want to know is, can these environmental exposures cause cancer, in which case, just like with genes, we can use these high-risk studies. So this is kind of where we work, where you have high-risk, high-susceptible individuals, to show that these environmental chemicals matter for cancer risk. So it's not to say that they only matter for people with a family history. It's just you're more likely to detect a signal in younger cohorts, and I'll show you a little bit more there. So recently we estimated sort of here, this would be a typical cohort. So a typical cohort recruits people where most of the cancers occur. So most of the cancers occur over here in your 60s, 70s, and 80s. And unless you specifically recruit young people, what happens is you miss a lot of people. In fact, you miss the most highly susceptible people. One, because you're missing those that got diagnosed very early on, because most human studies will exclude people who've had prior cancers. And further, you're missing people who are highly exposed. So you can imagine, if we're trying to understand the effects of air pollution or the effects of some chemical that people are highly exposed to, they may not be included in your cancer cohort because they may have gotten some other kinds of diseases, chronic diseases in midlife. So you're missing the highly susceptible and you're missing the highly exposed. Furthermore, for breast cancer, you're missing people outside of every single window of susceptibility. So unless you have people around the menopausal transition, you're measuring environmental exposures and you're measuring the breast tissue when most of the dynamic activity and the metabolic activity is finished. So again, just because we have 100 studies doesn't mean that we've asked and answered this question. So in our high-risk cohort, These are all individuals who were recruited, again, primarily to find BRCA3, which there isn't one, as everyone knows, but there's many other genes that are important to breast cancer, but at a much lower prevalence. You can see that there's a range of absolute risk. So these high-risk cohorts, I would argue, have a lot of advantage. One, because they have sufficient statistical power for this tail of the distribution. Further, they have plenty of people that are what we would call average risk. So how do we calculate that? If we take the 1 in 8 lifetime risk, there's thousands of people in our so-called high-risk cohort that are average risk. So again, an important thing to recognize is that even though you have a family history itself is a poor discriminator of your absolute risk, because it all depends on when your relatives were diagnosed and the genes that may have been present, and whether or not some people have relatives that were diagnosed with cancer very early on but have no germline mutation, of which that wouldn't then lead to an increased risk for you because they may have had their cancer from a somatic mutation. And what you can see here in this schematic is this blue bar here represents sort of average risk cohorts. So most of the cancer cohorts that are, in the evidence base, so to speak, are what we call average risk. So they're not recruited through their family history. So there's plenty of statistical power to look at how exposures affect average risk, but there's very low power to understand gene environment interactions. And if you're trying to study environmental exposures that everyone's essentially exposed to through their water or their soil or their air or their food or their plastics, then it's very hard to measure. And if people are not highly susceptible, you get a lot of inconsistencies. So we did the same sort of review that Dr. Brody and her colleagues did. Again, on the left-hand side are similar groupings of chemical compounds. And here across the different matrix is really to say how many studies. And just like they found for very few studies are done in windows of susceptibility, most studies are not done with these enriched cohorts. So in fact, there's 2 family cohorts in the world that have recruited family members. One is this breast cancer family registry that we run. The others is the sister study. I think you heard more about the sister study last month. And then also there's studies that have looked at early onset cancer, so cancers under 50, or G by E interactions. But again, the vast majority of studies don't enrich for any of these 3 things. However, in the studies that did, we saw, again, much greater consistency. Over 75% of the studies were positive in different kinds of compounds. So again, the zeros here represent that there's still a lot of gaps for certain chemicals, but we're hoping to fill those within the next year. So when we look at this gene environment interaction a little bit closer, on the x-axis, again, is your absolute risk of breast cancer. And then these are different levels. In this case, it was polycyclic aromatic hydrocarbons measured objectively through a blood biomarker. And what you can see is the people who had a lot of exposure, so in the 90th percentile, and also a lot of absolute risk, a lot of susceptibility. This is based on their family pedigree. So not just how many relatives they have, but what were the ages of their relatives when their relatives got cancer. And in this case, we do a complete pedigree, so it includes second generation, which is important, again, because genes like BRCA1 or 2 can travel through the paternal line. So it's very important to do second generation to ascertain risk as well. And you can see these odds ratios are very high, so much so that, again, with high exposure and high susceptibility, this is a zone where, even though these are not randomized clinical trials, again, though, for the most part, there's no cancer cause that's been tested in a clinical trial unless it's a pill-based chemoprevention. What you can see is they're in a zone that is unlikely to be explained away by bias. So we think that's important. And then last month, we finally finished an analysis looking at the full breast cancer family registry. So this had a lot more statistical power. And what you can see here on the left-hand side is a replication of that finding, where green here is representing the people who are highly exposed, and under 50, you get this very strong gene environment interaction, again, where the relative ratios here are very high that can't be explained away by bias. Whereas over here, over 50, you get, still a little bit of a gene environment interaction, but you can imagine if this were done in an average risk cohort, probably most of these confidence limits would not include, would probably include 1, in which case this is, again, back to this, oh, we've tested environmental chemicals in hundreds of studies and there's no consistent message. Here, I would argue that once you look at people who are highly susceptible, just like we try to find genes in people who are highly susceptible, those genes still matter for people over 50. And I would argue those environmental factors still are, of course, are just as important for people over 50. It's just that it's harder to detect those signals. And so, again, in thinking about the implications, I would argue that we really need to think about this underlying susceptibility. And just because the study is published doesn't mean that it's addressed susceptibility. In fact, it's much more common for most studies that are published to not address susceptibility. And then in the case of environmental exposures and breast cancer, thinking about genetic susceptibility is very similar to thinking about windows of susceptibility, in that you get a more consistent message. And just like with genes, results from these enriched cohorts are still relevant to those later in life and without a family history. So I want to just spend a couple minutes now on thinking about screening, because this is really important, particularly now that we know that breast cancer is increasing under 40 in the US, and as well is increasing globally under 50. And again, most countries outside of the US do not screen until 50. Most countries don't have population-based screening programs unless you're looking at just the high-income countries. Some of the high middle-income countries do, but most of the lower middle-income countries do not have population-based screenings. Countries that are have high population density but have a lot of rural areas, just like in the US, our rural states really don't have great capture of screening either. These are very important things to think about for scalability now. So what if we can think then about how then to reach these individuals who are higher risk? Because family history is not going to do it if these trends are increasing, because family history has always been used to proxy for germline genetics, even though we know it's much more than that, and we know that it's also shared environment. But nevertheless, how will we find individuals that don't have a family history that increasingly are being diagnosed with early onset cancer? So can we think of a hybrid between clinical breast exam and population-based genetics with phenotypic biomarkers? And some of those biomarkers may prove in the future to be environmental exposomic markers. Again, the early days on that, but it's still a possibility. Here you can see different countries that are already using this kind of model, where community health workers are going out and doing clinical breast exams. So the question is, how could we combine sort of a clinical breast exam, or should we go back in the US like to a mammogram at age 30 or 35 to be a baseline mammogram? Again, these are all things to be discussed and weighed, but we can't just rely on family history. Now, one of the things that's still a challenge is that guidelines for genetic testing, clinical guidelines, mainly rely on family history. So again, this is a challenge because one, if the cancers that are increasing are not because of a family history, and a close friend of mine, she was diagnosed at 30, and then her mother was diagnosed in her late 60s after her daughter was diagnosed. So yes, her mother then had a family history. But again, the increase in these early onset cancers are really happening younger and younger, and without people that necessarily got genetic testing because they didn't have a family history. So again, how can we think about the implications and the cost? So I would argue we need to think about this collectively now because of the cancer incident trends, which include both in the US, but certainly globally. And also the fact that we know more and more, given these increasing trends, that there's going to be a mismatch between our clinical guidelines and who's at risk. And then further, there's this inequality or inequity in terms of who even gets genetic screening. And then finally, there's more and more data on cost effectiveness of population-based testing, which if we have any time during the question and answer, we can talk a little bit more about that. So back to triple negative breast cancer. So again, one of the most aggressive breast cancer types that we have. We know, and you saw some of the data earlier, that across all age groups, but again, especially early onset, but throughout life, non-Hispanic Black women have double the rate of triple negative breast cancer than non-Hispanic white women. And what we have found out is that the gene prevalence for triple negative, the prevalence is the same in these women. So again, the genes can't explain the doubling. So, but what can explain the doubling is how those genes interact with the environment. So the problem is who gets genetic testing. This is sort of a flow on the left of what we have in this country, which is you don't really get genetic testing until you're identified to have a family history. And you can see there's just so many places that you can miss out. And in particular, we did some scenarios and sensitivity analyses just to see how many people would miss out. And again, you can see the much bigger burden or inequity that exists now with genetic screening is because non-Hispanic Black women are being missed because of not having the family history, but still having the same prevalence in these genes. So again, if we could think about doing either a baseline mammogram at age 30 or 35, and then also combining it with now in many clinics, people are using polygenic risk scores in combination with the intermediate and high penetrant breast cancer genes. You can see here, and oops, so sorry, across the board, that some of these markers, and this was something we just did using our hospital data, do help discriminate absolute risk more than family history or just as well as family history. So why not offer this test, which could be easily done when someone's 25 or 30, just so you understand, again, their risk that won't change based on germline genetics. And then I would argue further, can we complement that polygenic risk score with what I call this geographic risk score? And again, you can see here that on the x-axis is what happened before 2010, on the y-axis is what happened after 2010. And these are incidences in the 50 states of breast cancer under 40. So you can see most states are increasing over time, but the absolute burden is still pretty much in the Northeast with lower amounts here in the West. So can you, this is just the first step, but can we zoom in then with communities, and particularly communities that are affected by exposures that we know increase risk, and some people at Washington University have done some interesting work with PFAS and early onset breast cancer. So can you zoom in based on community and do more screening in those communities along with polygenic risk scores? So I want to just speak for 1 or 2 minutes about treatment. And again, this is an area that hasn't been very well developed, but certainly one that we want to lead on now at Silent Spring, and an area of investigation that I think is really important for thinking about how environmental chemicals may interfere with treatment. And this is important because we have a lot of treatment for breast cancer, but again, the outcomes based on treatment are not all the same. And so we've learned a lot of things, particularly from COVID, but certainly now more and more from lung cancer outcomes, is that air pollution, neighborhood air pollution itself, people who are on immunotherapy do worse. So can we, extend that to look at, again, people with triple negative breast cancer? Some of them are treated with immunotherapies. Can we look at that with other kinds of chemicals, including air pollution, but other chemicals that influence inflammation and might affect outcomes with immunotherapy? Can we look at some of the endocrine disrupting chemicals with respect to endocrine treatment? One example I'll just show you that again relates to the most aggressive type of breast cancer, which is triple negative. This was a study that was published by a group who looked at just antimicrobials. And so it may be hard to see all these survival curves, but suffice it to say, this study was so important that now the recommendation is, if you're being treated with triple negative, to not use antimicrobials. So again, this is an area that we want to examine rigorously with other kinds of exposures, because if that can improve outcomes after breast cancer, that will be important too for understanding how chemicals can affect progression with cancer. So finally, again, I would argue that thinking about environmental chemicals and where we can make inroads in terms of what we know and how to improve risk assessment and prevention across the cancer control continuum, I would argue, again, these higher risk cohorts can provide evidence that can be used then for population-based interventions. And we've been planning a few population-based interventions in households through some of our programs at Silent Spring, but we also hope to do more of those kind of interventions with reducing environmental exposures. And then also, as I mentioned earlier, hopefully we'll see a day where a panel of blood biomarkers, including phenotypic markers that relate to DNA repair, can be used in combination with some of these genetic risk factors for population-based early detection. And then finally, as I mentioned, we hope to do more studies in this space looking at how environmental chemicals affect treatment throughout. So in the last few minutes, I do want to say sort of where we'd like to go to in thinking about why these chemical exposures may relate to some of the differences we're seeing with these 2-time difference in triple negative in non-Hispanic Black women compared to white. And so colleagues at Silent Spring, led by Dr. Kay, have published last year a very important list of breast cancer relevant chemicals. More recently, the group, we've looked at that in terms of commonly used plastics, and of the list of over 900 chemicals that have been shown to be either genotoxic or again, disrupting different endocrine pathways. Over 400 of these chemicals are in everyday plastics that we're all exposed to. So again, we want to kind of look at where that relates to. And the group at Michigan, and again, these are early days, but have published studies that have shown some of these chemicals may actually affect cell plasticity. So may actually change luminal cells to basal cells. So may explain why some molecular subtypes may be more common in people that are heavily exposed to these chemicals. So in the end, I just, again, I'm so thrilled to be speaking with each of you, because MBCC really has been the leader in advocacy and in developing social capital within communities. So I want to end with that because I also want to just summarize some of the work I've been doing over the last 5 years in building some of these networks. And I hope we can all work together in building networks even further across the country, because this is a time we really need to come together more, and we can't rely necessarily on these networks being funded through the federal government for us. So let's find other ways to do it. So I'm part of a great privilege of one grant that didn't get canceled. It's called the SOCA grant, which is Social Capital Promoting Multi-Generational Health. And as I showed for breast cancer in particular, multi-generational health is really critical for thinking about those windows of susceptibility. This is happens to be the CEO of the largest healthcare system in New York State, Michael Dowling, who's actually a social worker and grew up in a very class-based society without indoor plumbing in Ireland, and now is the CEO of, as I mentioned, the biggest medical system in New York State. And during the pandemic, he said, "This is who we are." And again, that resonates a lot with me because he overcame so many different barriers and so many class divisions. And really, we've been trying to do the same, because there's no way we're going to solve issues around environmental injustice and breast cancer inequities and all of these without partnerships. So this is some of the partnerships I've been growing over the last 5 years with colleagues at different institutes. Unfortunately, this is one of the ones that was canceled, but it did help us, based on these collaborations, build this SOCA Center. And I want to end with one of the things we've been doing over the past year with thinking about how to build social capital. And so building social capital, again, helps if you have financial capital, but it is not by any means necessary that you have financial capital to build more social capital. And that's something we can all do together. And so this is the group I'm still working with now for this center. And these are all the community partners that we work, including school-based programs, because again, the group that's most affected by everything going on right now are really the young people. So what can we do in the schools, and what can we think about in terms of using all the networks and influence we have to help the generation right now that needs to know what is known about environmental health. So again, social capital is about building networks, engaging people, building educational programs, and help with strengthening bonds. And this is exactly what the MBCC does. So again, I'm really privileged to be able to start working with you and thinking about things we can do together. And so again, with working together, we really want to think about, what is the right network to build? Is it bonding networks? Is it bridging networks, linking networks? All of these are very important for building social capital, but in different times. And so again, for each problem we're trying to tackle, whether it's cleaning up the water or getting information out to high school kids, we have to think about the networks and how we can most help in this space. So, and all of the components of social capital involve trust, engagement, connections, and networks. And so some of the work we've done recently in the Bronx related to thinking about bonding networks, whereas work we were doing in Washington Heights, they were more interested in linking networks and connecting people across the different organizations. So just to give you an example quickly of what we ask each group when we start working with them is really what's most important to them in terms of if you could strengthen residents' trust and engagement in one of these kinds of networks. So we go through each of these, because in different times of engagement, different bonding, linking, and bridging networks can be most relevant. And then we go through these exercises of, how would you measure success? What would be important for you? How to build trust? And so we've done this, and across, again, a variety of grants, some canceled, some not, but I would argue that all research really needs to be done alongside these community programs, training the next generation, and having ways for community partners to lead in these initiatives. And we have done grant writing workshops and love to continue any of these programs in Massachusetts. And so we are starting the first, and this is where I'll end, the first ever high school program that, it's not the first ever, but the first one we're all doing here this June, and really hope that you can spread this message widely. And really what we hope to do is fill that vacuum of information about what we know for environmental health, and really give high school students an opportunity to network and to think about doing careers in research. And with that, I will stop and take some questions. So thank you all.
Thank you so much, Dr. Terry. I would like to invite any of our folks who are on the webinar to submit any questions they may have by going to their GoToWebinar panel. And there is an arrow that indicates questions. If you click on that arrow, a field will open up and you can submit your questions that way. While people are submitting their questions, Dr. Terry, so could you touched on this a little bit in your webinar, What are some reasons why early onset breast cancer rates may be going up?
Yeah, that's the million-dollar question. And, we do know that it can't really be explained by obesity, where some of the GI cancers can, because early onset breast cancer isn't really obesity driven. And again, the data that we've looked at both within the US and also globally suggests that it can't be fertility patterns or changes in childbearing. So, we do know that there's a lot of differences in environmental conditions, a lot more use of plastics within this generation. And those are the things that we hope to look at more rigorously with some of these cohorts that we have right now.
And you mentioned that there were some objective measures of increased susceptibility, such as breast density. Are those used in any proactive or preventative way to kind of think about like initial screenings, any of those that are not necessarily related specifically to family history?
Yeah, it's a great question. So, the measures that we use in our research studies with the optical spectroscopy, that is not FDA approved, so that's not used clinically. Clinically, you can still use MRIs or mammograms, but again, most people wouldn't have access to MRIs unless they're already deemed high risk, so that's a challenge. So again, this is a challenge that we all have right now, which is how can we screen for high risk individuals? Should it be some combination of communities that we know are at risk combined with genetics, combined with clinical breast exams, or, again, should we go back to a baseline mammogram at 35? There's a lot of questions to think about.
And what do we know about why the rates of the most aggressive breast cancers vary so much by race and ethnicity?
Yeah, that's a great question too. Again, I think that's also a million-dollar question. We have a lot of programs going on right now at Silent Spring looking at, report back of environmental chemicals that are in products that personal care products that are used in women of color that are more frequently used there. We do know that there's growing data about air pollution and its association with breast cancer. So we do know environmentally that that communities are not all at equal risk.
One of our attendees just submitted a question regarding Hispanic, right? We usually say white, non-Hispanic, or Hispanic is kind of a demographic indicator. Why is Hispanic separated and pulled out? And does it include those who have their heritage is from Spain, which those folks may identify as white?
Yeah, it's a great question. I mean, most people will say that these demarcations that are used in a lot of registry data that may not be used anymore, are imperfect because they're very heterogeneous. I do know that there is a very different breast cancer risk for people from the Caribbean as opposed to people from Mexico. So, when you look at US data, the majority will probably be related to people from Mexican descent. So, there's a lot more that needs to be looked at by that. But, the thought with the higher risk in Caribbean women from Caribbean areas is really from some of the genetic admixture as well as the environmental exposures that are part of many of these island communities. Yeah.
We know that screenings are really, fundamental to, identifying breast cancer and early onset, right? Like it does save lives. We've seen that data that when we know that people are being screened earlier, they are, in fact, we can map those changes in the breast over time and identify breast cancer at an earlier stage. So what are the strengths and challenges? What are the advantages and disadvantages of our current system of breast cancer screening?
Yeah, I think the biggest disadvantage is it's based purely on age rather than risk. Also, mammography itself is not as sensitive in premenopausal women as it is in postmenopausal women. So, it's not a perfect screen either. I think what's really important, given the increase in early onset cancers, not just breast, but all cancers, is that people do have a better understanding of changes in their body, and they should go and seek healthcare when they see changes in their body, and not be turned off if someone says you're too young to get cancer, because that is, the common misperception that a lot of people may say is you're too young to get cancer. And, people know their own bodies, and they should go and seek healthcare.
And so self-exam is probably a very important tool in the toolkit for that self-identification and knowing one's own body and noticing when there is a change. Would you agree with that?
Yeah, so self-exam, is not part of clinical guidelines. And that was because it was a very large randomized clinical trial from China where they didn't show that self-breast exam worked. However, my colleague, Dr. Houghton, has done a lot of work with early onset patients, and, that's primarily how a lot of early onset patients have found their cancer. Either they or their partner have felt palpable tumors. So, I think clinical guidelines say not to do it, but again, I think people know their own bodies and should seek healthcare if something is unusual.
And I know we're a little bit past the hour. Why are key unanswered questions regarding the role of the environment in outcomes after breast cancer? So we have someone who's been diagnosed, they've gone through their breast cancer journey, now they're being monitored over the rest of their lifetime. Why aren't we monitoring the impact of environmental outcomes for that cohort of people?
Yeah, I mean, we haven't yet, but again, we're launching a few studies to do that this year. I do think it's a big unanswered question, and I do think based on studies that have been published, for example, with air pollution and lung cancer outcomes, I do think it's an important question generally that needs to be asked and answered.
Dr. Terry, thank you so much. On behalf of myself and the Massachusetts Breast Cancer Coalition Board and Cheryl Osimo, our Executive Director, I want to thank you for this very informative discussion today. And I want to thank all of our many listeners for joining us. For those interested, the recording of this webinar will be made available later today on the MBCC website at mbcc.org. And if you would like to further support our mission of preventing environmental causes of breast cancer, please consider making a donation by visiting mbcc.org/donate. I appreciate your consideration and have a very good afternoon.
Thank you so much.